Hello everyone. The next topic which we will be discussing is about the centrioles. So, what is a centriole and what is the role of centriole in a cell? So, as per the standard definition, centriole is an organelle which is cylindrical in shape and it is composed of a protein which is called as the tubulin. The main function of centriole in the cell is the centrioles, they are concerned with the cell division. What they do is they help in the formation of the spindle fibers and they separate the chromosomes during the cell cycle. So as I have told, they are cell organelles that are cylindrical in shape. Now let's start with the introduction first, that all these animal cells have two centrioles. They help the cell during the cell division process. They work during the process of mitosis, that is the equational division, and the meiosis. They could be found in some lower plants such as Calamidonaz, although they are not present in many of the fungi, angiosperms, and the conifers. They are usually present near the nucleus, but they are not visible when the cell is not dividing. So this is the basic things that what is a centriole. Now let's discuss that what do centrioles do and where the centrioles are found. So centrioles, they are cylindrical cell structures that are composed of grouping of the microtubules, and they are which are basically tube shaped molecules or strands of the protein. So without the centrioles, chromosome will not be able to move during the formation of the new cell. So this is the main function of the centriole, that without the centriole, the chromosome will not able to move during the formation of the new cell. Centrioles, they help to organize the assembly of the microtubules during the cell division. To put it simply, chromosomes use the centriole microtubules as a highway during the cell division process. So this is about the centrioles. Now coming to the structure of the centrioles. So all the centrioles, they are formed of 9 groups of microtubule triplets that are organized in the cylindrical shape. So this is a microtubule triplet that is being shown in the diagram, and it is of what shape? It is of cylindrical shape. The detailed structure of centriole can be divided or can be studied only under the electron microscope. So they can only be studied under the electron microscope. They are associated together at right angle to each other. So they are associated together at right angles to each other. Now, this is the distal appendages that are being shown here. This is a mother centriole and this is a daughter centriole. They have proximal ends, they have interconnecting fibers, they have distal ends, and they have appendages as well. So the question is that who discovered this centriole? So that was Edward van Benden and Boveri, who observed and identified the centrioles for the first time in 1883 and 1888. The structure of duplication of the centrioles was first given by Joseph G. Gall and Harvin in the 1950. So centrioles, they help in organizing the mitotic spindles and the process of the cytokinesis. The centrioles were believed to be necessary for the formation of spindles, for the mitotic spindle in case of the animal cells. Although there were recent types of research which have explained that the cell which does not have a centriole can function without it in the G1 or interphase, and they can be formed later in a de novo manner. So this was the recent research. Now, the location of the centriole plays a key role in the three-dimensional organization of the cell, as it also regulates the location of the nucleus. In flagellated and ciliated organisms, the location of such organelles is decided after the mother centriole that forms the base. So it is here the role of mother centriole comes in the case of ciliated or flagellated organisms. Next is, where are these centrioles found? So if we talk about this, then centrioles, they are found in all the animal cells and only a few species of lower plant cells. So the two centrioles, that is a mother centriole that was being depicted in the diagram, as well as in case of the daughter centrioles, they are found within the cell in a structure which is called as the centriome. Now, what is the composition of the centrioles? So most of the centrioles, they are made up of 9 set of microtubule triplets, as it was been depicted in the diagram as well, with the exception of some species such as crabs, which have 9 sets of microtubule doublets. And there are few other species that deviate from the standard centriole structure, and microtubules, they are composed of a single type of globular protein which is called as the tubulin. So next is about the functions of the centriole. Now, what are the functions of the centriole? So these are some of the important functions of the centriole. In spite of being devoid of DNA, the centrioles, they are capable of forming new centrioles. So they are capable of forming the new centrioles. So they do not have their DNA, but they can make their own copies, or they can make their own new centrioles. They can be transformed into the basal bodies. The basal bodies will give rise to cilia and the flagella. They help in the cell division by forming the microtubules organizing centers. And out of the two centrioles, the distal centriole, they forms the tail and the axial filament. So this is what is about the function of the centriole. So if we want to brief this, then during the mitosis of or cell division, the centrosome and the centrioles, they replicate and they migrate to opposite ends of the cell, as it is being depicted in the cell division part itself, that they replicate and they migrate to the opposite ends of the cell. Now what the centriole is, the centriole, they help to arrange the microtubules that move towards the chromosome during the cell division to ensure each daughter cell receives the appropriate number of the chromosomes. Now centrioles, they are important for the formation of the cell structures, as it is also been depicted in this is that give rise to cilia and the flagella. So cilia and the flagella, they are found on the outside surface of the cells. They aid in cellular movement, and a centriole combined with several additional protein structures that is modified to become the basal body. And basal body, they are anchoring sites for moving cilia and the flagella. So this is what are the functions of the centriole. Now what is the role of centriole in the cell division? So centrioles, they are located outside of but near the cell nucleus. So centrioles, they are located near the cell nucleus. In case of cell division, there are several processes in order of their occurrence. For example, first is there is interphase, then comes the prophase, then metaphase, anaphase and telophase, in which the two daughter nucleus are formed. Centrioles, they have very important role to play in all these phases of the cell division. Here the end goal is in moving replicated chromosomes into a newly created cell. So this is what the role of centrioles in the cell division. So this is the important role in the cell division. Now if we want to sum up all of this, then centrioles, they are cylindrical in shape. They are paired organelles that are cylindrical in shape, and they are found as single structures in the cilia and the flagella in animal cells and some of the lower plant cells as well. They are constructed of the microtubules, and in animal cells there are centrioles that organize the pericentriolar material to produce microtubules that includes the mitotic spindle fibers. Now this was all about the centro. This was all about the centrioles. Then these are some of the references.